I am Kika Sucharov and one of the co-chairs of the BCPS Young Investigator Award Committee. And I am here today with Dr. Liu, who is one of the finalists and is going to tell us about his work.
Yeah, happy to be here. Thanks for the opportunity. And yeah, right now my name is Shijie Liu. I'm an assistant professor in Cincinnati Children's Hospital. So I just started my lab in 2023. So my lab is about 2.5 years old. So I'm happy to share our most recent study about cardiomyocyte proliferation and also about what we think is the most important thing for cytoscatter in the cardiomyocyte. We try to understand the mechanism, how this sarcomere structure be regulated, and why this is important for cardiomyocyte proliferation.
And what are your findings on that?
Yeah, our finding is because we know adult cardiomyocytes have very limited self-renewal ability. That's the main reason for the cardiac disease, which is the leading cause of death worldwide, because the cell has very limited ability. So basically, after injury, the cardiomyocyte, which is cardiac muscle cells, they cannot be replenished. So recently, we find these adult cardiomyocytes, they can proliferate and division, but the ability is very limited. So based on the last 2 decades of study, we find some factors like HIP pathway, ERBB, and the microRNAs that can stimulate adult cardiomyocyte proliferation. And during this process, we noticed that these adult cardiomyocytes, they have to de-differentiation, go back to a fetal-like state, and then accompanied with a sarcomere breakdown, and then they can overcome the structural block and divide. So there's very limited knowledge about how this cardiomyocyte de-differentiation and how this structure be regulated. So we are really interested in what kind of factor regulated this structure reorganization. And our finding is that when we stimulate the cardiomyocyte proliferation, we find significantly increase the microtubule network, which is required for the sarcomere structure disassembly. And also we find that there's a direct physical interaction between microtubule network with the sarcomere structure, which facilitate the sarcomere structure continue breakdown.
And what about the reassemble?
Yeah, that's a good question. And we think this is a redifferentiation process. So far we have very limited study about this part. And luckily our model we are using, we can control the factor expression. Basically we can turn on and turn off. So right now we just turn on and we study all this process. And then when we turn it off, the cardiomyocyte go back very quickly. But in this process, we haven't really studied what kind of molecular mechanism there. But we have this model. I think in the future, we can definitely do that. So right now, I want to say we really focus on the first phase, just how these cells enter the cell cycle and how they disorganize the structure. I think this is very important. And we also think the cell cycle progression and this sarcomere disorganization, they may independent to each other. So basically, when we turn on the process, all these processes, they're ongoing together or at different time, but only they align perfectly in one cell, this cell can successfully divide. So I think still there's a lot of unknown, and in the future, if we know better about all these different mechanisms, we can better manipulate all this process, maybe make the cell divide more efficiently and also make the function better.
Do you think that there are some cells that are going to be more prone to de-differentiating and dividing, or is this a very uniform process in the heart?
I think so far we think this potentially will be a uniform process. Okay, we found in all different factors that stimulate cardiomyocyte proliferation. They also notice this disassembly of structure. They also notice cardiomyocyte differentiation. Yeah, this process looks like very, very uniform in all these different models. Yeah.
Very interesting.
Thank you.
Yes.
Yeah.
Well, we are looking forward to your talk tomorrow. Good luck with everything.
Thank you.